All right, I'd like to start off thanking the organizers for inviting me here. This is a really cool full circle moment for me because back in 2014 when the MinION was first in pre-release, I submitted a horribly written application to try and get my hands on one as an undergrad. Did not get one, and here I stand now talking about genomes that I've assembled with PromethION data. So it's a really cool full circle moment for me. So the first question that I get a lot of the time is, why the heck are you talking to me about marsupials? And it's really because they're an amazing model that substitutes for mammals in a lot of ways, because they're very closely related to mammals, but they have a lot of really unique phenotypes that differentiate them from mammals that we can use to compare them. So they have a very unique method of locomotion. The unique reproduction is very heavily focused on. And some of the most dynamic forms of cancer that exist on the planet are in marsupials, specifically the Tasmanian devil. So they have the transmissible form of facial tumors that contact transmit between individuals. So we can start to study this with the rapid genomics that we're able to do now that Oxford Nanopore has advanced so much. So we have these genomes that we can just churn out now with these R10 reads that have the much higher throughput. So putting this into a little bit more perspective than I did yesterday, we're able to look at deep comparisons between the lineages, where in my thesis I focused on macropods, which are the wallabies and kangaroos. And thanks to collaborations with Colossal Biosciences and Andrew Pask's lab in Australia, we're now adding dasyurids, which are carnivorous marsupials, to the project, as well as hopefully some of these really cute guys like the greater bilby, who I love in the near future, hopefully. The story that I want to tell is about the genomic diversity that complements this phenotypic diversity, though, because this has been the focus of my thesis. So we have these really cool examples of karyotypic diversity in the carnivorous marsupials, like the dasyurid family, where there's 20 million years of evolution, and the genomes are pretty static. You can see that there's a little bit of movement of these blocks that have been mapped with fluorescence and C2 hybridization that are well known across all marsupials. But if you compare them to kangaroos and wallabies, these are two very closely related species, the Tammar wallaby and the swamp wallaby. The chromosomes are completely different. You can still identify those blocks, but they're in a totally different order. And the swamp wallaby actually has a completely different sex chromosome system. They have an X, Y1, Y2 system, and they're the only kangaroo or wallaby that does. So that's the one on the right side of the slide here. So I set out in my thesis to answer the question, why is this happening? What's going on in these genomes that allows this karyotypic flux, but they still end up with a functional genome? And the rapid advance of both ONT technology and all the inspiration that came from the telomere-to-telomere consortium allowed us to start to put together these genomes very, very accurately and very completely, to the point that we're able to put them together very well with just 2 flow cells most of the time. So we can get a ton of accuracy from a duplex run with the standard ligation kit, the version 114, along with some ultra-long reads, and then some high-C, omni-C, poor-C to scaffold them together. And I have to credit Nicole, our sequencing queen, for all our help putting these libraries together, because she's absolutely incredible at generating this data. We're getting ultra-long runs in the N50 of 160 kb range with quality scores in the mid-20s, which give us unbelievable genomes to work with. So the genomes that I'm about to show you were all assembled with a mix of Fly and the Virco assemblers, where we input duplex sequencing instead of PacBio reads, and compared them between 2 individuals. So I have a male and a female of each of the species that I'm talking about, with the exception of the swamp wallaby, so that we could compare them across individuals to get these genomes structurally sound. So stats for the other genome dorks in the room, don't get too overwhelmed with this. The point that I'm trying to make here is that we have quite a few levels of genome diversity. On the left side of the slide, I have my older R9 assemblies that came from my thesis. So these are the macropods, the Tammar wallaby and the swamp wallaby. both near T to T level, but the accuracy is a little bit lower if you look at the mercury and K-mer completeness at the bottom of the chart, because these are R9 reads. So that's just how it works. They're a little bit less accurate. And now we're moving into these newer R10 reads that we have beautiful genomes, but I still have a little bit more work to do. If you look at the Ns per 100 kb row, I still have some gaps to work on in these genomes. But we have some beautiful genomes that we're going to release in the near future to use as resources for the marsupial community. But what I'm really interested in is comparing these two groups. So doing whole genome alignments of these two groups, we can see that in my pet group, the macropods, the wallabies and kangaroos, we have all this flux, and we can see these when we align the whole genomes to one another. You can see that there's all these green strings all over the place where these synthetic blocks are just a big mishmash. But in the carnivorous marsupials, the dasyurids on the left with the blue strings, everything kind of stays in place. But that was 4 times the evolutionary distance. So what's going on at all these junctions? Well, it's a really cool answer, because when you start to dive into more details about direct comparisons between what's actually happening at these breakpoints, you can see that there's a couple of splits that I highlighted with black bars. If you line these up with the karyotypes, they're in a very specific location. These are all centromeres. So in the wallabies and kangaroos, the centromeres are all acting as these syntenic fusion and fission breakpoints. So something really weird is happening at the centromeres in wallabies and kangaroos in particular that cause their chromosomes to be in all kinds of states of flux. And luckily, thanks to ONT sequencing and specifically ultra-long sequencing, we were able to get linear assemblies of these centromeres so we could actually start to look at and answer questions about what's going on there, what's allowing that to happen. So when we start to look at the actual sequence there, and this is just chromosome 5 as an example because it's my favorite plot. This is a stained glass plot that happens to look the prettiest. So if you haven't seen a stained glass plot, more red is more similar. So if you see a big red triangle, very similar sequence. The more blue-green it gets, the less similar the sequence. We know that this is the centromere because we aligned CEMP-A here, which is the epigenetic marker for the centromere. And thanks to the ultra-long reads, we also have methylation data. So we can see and verify the methylation dip that occurs in the centromere that was also observed in CHM13, the CDR. But what's really amazing that we discovered here is that the sequence here is actually a simple repeat. A 6-letter simple repeat comprises the entire centromere, and it's this simple repeat. Does anybody recognize that? This is the telomeric simple repeat. So every single centromere, with the exception of the sex chromosomes in wallabies and kangaroos, is composed of the telomeric simple repeat. I was pretty shocked when I found this. My advisor, Rachel O'Neill, was like, you better be right. This better be a real thing. And we were able to verify this because there's actually been a lot of fish studies in the past that have noticed, hey, there's interstitial telomeric repeat in these genomes, but they couldn't tell that it was in the centromere. So what we think is going on is that because this simple repeat is here, it's enabling all these kinds of changes because it's able to bind to itself. And that's caused all these fusions where it's remained telomeric in the dasyurids. So we can also start to compare these across these genomes because in some places this is a centromere and in some places it's not, because we know where all these synthetic blocks are. In places where it's a centromere, it's retained its sequence identity. So in the Tammar wallaby on the right, where it's still a centromere, it has a very high sequence identity. It's maintained the identity of the telomeric simple repeat. And where it's no longer a centromere, it's lost it. So it's still recognizable, but you can see that it's blue. It's much, much more diverged from that simple repeat. Interestingly, it still has a methylation dip. I can't answer the question why yet, but we're looking forward to diving into that some more. But there appears to be a really interesting selective pressure to maintain that sequence at the centromeres. So we're really excited to keep diving into this because we can start to answer even more questions with these complete assemblies, looking at not just these fragmented reference genomes, but once we start mapping more data to these, we can see, well, what's missing in these hidden regions that we weren't able to see before. And we found all kinds of patterns that I'm really excited to be putting on in a paper in bioRxiv in the next couple of weeks. So keep an eye on Twitter. I'm very excited to release this paper. But we can learn all kinds of things about admixture and hybridization and what's important in repetitive elements and unique gene features, and hopefully learn more about these endangered species and learn more about these interesting things like the tumors in the Tasmanian devil and some other interesting species that we work on in Rachel O'Neill's lab, like deep sea corals that aren't explored much genomically at all. So just to sum up, all these technological advancements like R10 and the KIT 114 has allowed us to do these genomes very accurately and very quickly. They allow us to answer these amazing questions that I was barely able to touch on today. But we've found that a major split in marsupials is defined by these repeat arrays and cause this karyotypic flux. There's much more to that story, but of course I can't touch on that in a short talk. And comparative genomics is really powerful at this kind of resolution. So I'm really excited to see where this field goes in the future. So I have a lot of people to thank for this, especially Rachel O'Neill has been the most amazing advisor on the planet, along with all the marsupial community that I have to thank for me being able to be standing here giving this talk. And thank you all for listening.